adaptive immune system is really complicated, but even though it's complicated, and this whole process can take days to get this whole, somatic hypermutation, affinity maturation, isotype class switching, it takes 3 to 5 days for that to be complete. But once it's done, it's great. So the couple things that I think I could kind of maybe clear up a little bit is, during that 3 to 5 days when all this somatic hypermutation and then isotype switching is going on, we still have the innate immune system kind of working on the infection a little bit, and then we still have those low affinity but high valency IgMs that are trying to, make the best of the situation, just kind of keep things under control until those B cells finish their job, perfect that antibody. And once they perfect that antibody and they start making the mature forms, IgG, IgE, IgA, then that B cell becomes a plasma cell, right? And then that plasma cell just cranks out a million identical copies of this antibody, the antibody that it's making in order to fight the infection in the best way possible. So that somatic hypermutation, where we have successive generations of B cells getting better and better and better, higher and higher affinity at the FAB region, that's called affinity maturation. The daughter B cells that have lower affinity die by apoptosis. The high affinity B cells keep perfecting it till we get to exactly what we need. And then we have the class switching that changes the heavy chains to what we need. These two are the initial low affinity ones that are produced by the B cells early on. These are the three mature isotypes: IgG, IgE, and IgA. Okay, how does class switching occur? So we have kind of splicing, sort of like we did with RAG, slicing to kind of remove what we don't want and add in what we do want. Okay, so here's the classes of antibodies. IgG, most common antibody, monomer, and if we were betting people, 75% of the antibodies in the bloodstream are IgG. It can help with complement activation. It's great at opsonization. Also helps with neonatal immunity. So IgG, since it's a small monomer, can cross the placenta and do natural passive immunity. So mom's IgG antibodies can cross through the placenta into the baby. And so for the first 6 months of life approximately, babies have Fc receptors in their GI tract, and so they, IgG not only crosses the placenta, but it also can cross through breast milk. And so for the first 6 months, a baby drinking breast milk drinks, not just the milk, but mom's IgG antibodies. Those IgG antibodies can bind the Fc receptors in the neonatal gut, and they can be absorbed intact as antibodies and then help with natural passive immunity, kind of help to protect that baby from anything that the mom is immune to, until the baby's immune system can kind of get up and running on its own natural passive immunity. So that's why they say, that's one of the reasons why they say breastfeeding is so good, because it passively transfers mom's immunity to the baby until the baby's immune system gets going. After about 6 months, those Fc receptors are not expressed on the GI epithelium anymore, so the baby can't absorb that antibody intact and use it for their immune system. But the antibodies are still proteins, and so they can still be digested and used as energy for the baby. So breastfeeding immunologically becomes less and less important after 6 months of age, but it's still really good nutrition after that. Okay, IgG. IgM, early antibody, can be a pentamer in the blood or a monomer on the B cell surface. IgA is one of the mature forms, present mainly at mucosal surfaces, but there was a question right before break that can IgA be monomers and circulate in the blood? Yeah, they're about 15% of the monomers that are in the blood. IgA can also, even though it's a dimer, IgA can also, or a monomer, it can also pass through the placenta or pass through the breast milk, and so it can be helpful also for neonatal immunity. IgD is only an immature form like the IgM, and it's almost exclusively just a B cell receptor. It's almost exclusively just bound to the the mature, naive B cell, and you don't really find much IgD in the blood. IgE is the one that is important in allergies and in parasitic infections, and its Fc portion binds to basophils and mast cells and can help with allergic responses. Okay, and then this just kind of shows the activities of some of the different forms of antibodies as far as can they neutralize, can they opsonize, can they do ADCC, can they sensitize mast cells, can they activate complement, can they go across the epithelium, can they go across the placenta, can they go extravascularly, or are they present in the serum? So hopefully that'll at least give you some appreciation of the functions, various functions of the different forms of antibodies. Okay, so take home points. The somatic hypermutation happens first, and so you fix the FAB portion to be perfect, and then you fix the FC portion to be perfect, and that's kind of the main sequence. And helper T cells are really important. Remember, there's a big interplay between helper T cells and production of cytokines, and also some co-stimulatory molecules. So CD40 ligand is the main co-stimulatory signal in that whole process. And so there's a syndrome called that is CD40 ligand deficiency, and we'll talk about that in our immune deficiency lectures. And so this is an X-linked hyper IgM syndrome. So in a child with CD40 ligand deficiency can't go past IgM. So they can make IgM, but they don't go past IgM. They don't do the isotype class switching because of the deficiency in CD40 ligand. So they just have a bunch of IgM, but they can't go any farther than that. So they're less efficient in their humoral immune responses because they still have that low affinity binding with the IgM. Okay, let's see. Well, that's kind of redundant. I talked about that a lot. Okay, so remember that the early antibodies, IgD and IgM monomer, are membrane bound, and so those are the rough drafts. And then after the somatic hypermutation and isotype switching, then to a large extent the antibodies are soluble, so they're secreted. IgG, IgA, IgE, they're secreted. They go to the blood or to the mucosal surface, and they do their jobs. Okay, summary. Okay, so most of what happens in this humoral immune response is dependent on T cells. So we talked about that progression where the antigen presenting cell finds an antigen, processes it, gives it to the helper T cells. The helper T cells can stimulate the B cells to start going through the somatic hypermutation and affinity maturation and isotype switching, and then clonal expansion. So when it gets to the clonal expansion part, so it's already finished somatic hypermutation and affinity maturation, it's finished isotype switching, it becomes a plasma cell, and it starts cranking out, thousands of copies or millions of copies of identical antibodies that are just perfect. And that those plasma cells also clonally expand, so they make identical twins of themselves, and they don't mutate anymore after that. They just clonally expand, so there's a bunch of these identical twins of all these plasma cells. They're all making exactly the same antibody, and that leads to a really efficient humoral immune response. At the end of the process, when the bacteria or parasite or whatever is gone and there aren't antigens left over, most of the plasma cells end up dying by apoptosis, and we keep a few memory B cells in case we run into that same infection again. Yes?
When you do the isotype switching, is it possible that you, like with the cytokines that get signaled, you wind up with like multiple isotypes?
Yes. So the question is, can you have multiple isotypes for one infection? So you might have one plasma cell making primarily IgA to go up to the surface, but you might have another one that's responding to the same antigen but makes IgG, so it kind of covers what's going on in the bloodstream and the organs instead. Good questions. Okay, so again, all this is happening in the germinal center, and that's why when we get a humoral immune response going and we have all those daughter cells being made with somatic hypermutation, and then we have that clonal proliferation of the mature plasma cells, that little follicle in the secondary lymphoid organ becomes great big and it becomes a germinal center. And it's called germinal because you see a lot of mitotic figures, a lot of activity in there. Okay, so we have sort of a wave of responses in the humoral immune system. So we have a primary immune response, the B cell gets exposed to an antigen in the secondary lymphoid tissue. It starts putting out the immature form IgM pentamers released from those B cells that have just encountered their bad boy antigen for the first time. And so that's kind of the primary immune response. So there's this little bump right here where there's a primary immune response, and this happens pretty quickly because the B cell can make IgM pretty quickly, but it's not really happy with the affinity of the FAB portion, and so it starts the somatic hypermutation and the affinity maturation, but that can take days. And so remember when we were talking about the adaptive immune system, it's slower to respond. It might take 2, 3, 5 days in order to go through the whole somatic hypermutation and isotype switching. But once it does that, it's awesome. It's really good. So you have this initial bump, the IgM bump, and then a little bit later you get the IgG or IgA or IgE bump when all of the tweaking is done. Okay, so we have, like over here, gosh, I keep going, I am just neglecting you guys. Such a terrible grandmother. So we have a little IgM bump, and that's that primary immune response, and then we start getting an IgG bump, and this is first time we've been exposed to that antigen, and so we get a pretty nice little IgG or a mature antibody bump. But then remember, those memory cells are kind of squirreled away and waiting for the second exposure to that same antigen. So if we get, like, let's say we get strep throat, we get an IgM bump, we get an IgG bump, we recover from the strep throat, but those memory cells are just waiting. And then a month later, we get another strep infection. But those memory cells don't have to go back through all of that somatic hypermutation and isotype switching and everything. It's got it already. So you can just leap into action. So look what happens the second time. You might still get a little IgM bump, but the IgG just goes through the roof because it already knows what to do. So the memory of the adaptive immune response is just huge, and it's brilliant. This is also, I'm glad I put this down here, it's also the rationale behind booster vaccines. So you know how you go and get a shot, and then they say, 3 weeks or 6 months, whatever later, you have to go get a booster shot, a booster vaccine. And that is taking advantage of this very phenomenon that, the first time you're exposed to the vaccine, you get a pretty nice little IgG response, you make the memory cells, but then you get a booster, and then, wow, the immune system just goes through the roof. And then you get even more memory cells, so you have even more efficient immune protection against that, bad guy. And also it's a faster response too. Okay, so this is kind of putting that all together. The IgM bump is kind of about the same, no matter how many times you get exposed to the antigen, but it's the IgG response that is much more dramatic with subsequent exposures to the antigen because of those memory cells. Okay, so real quickly, we talked about most of this already, so this should make a lot of sense. What do these antibodies do? They can neutralize, they can opsonize, they can activate complement, they can activate ADCC. So this is the, and these are monomers, but I kind of wish they were dimers right now. So this is the mucosal surface. These are, pretend they're dimers, IgA dimers, and they're on the mucosal surface, and they're gonna bind that pathogen and neutralize it so that it never really gets into the gut or into the respiratory system. So that's pathogen neutralization. Doesn't really destroy it, it just grabs onto it and won't let it invade and do its job. And then if it's in the gut, it's just gonna get digested and washed out. Okay. Antibodies can also activate natural killer cells. So natural killer cells have Fc receptors, and they can bind the antibody that has bound the pathogen. And then remember, natural killer cells use perforin and granzyme. So the natural killer cell with its Fc receptor binds to the Fc portion of the antibody that's bound to its epitope, and then the NK cell can kill that, that tumor cell or that pathogen, and the target cell dies by apoptosis. So this is really great with cancer. Okay, remember antibodies can also activate the classical complement system, and it shows IgM, IgM pentamer up there that can activate the complement system. And that's one of the other things, while the somatic hypermutation, affinity maturation, isotype switching is going on, the IgM, not only do they have really good valency to bind a bunch of pathogens all at once, but it can also activate that classical complement system and kind of get that going to help to knock down the infection too. And of course, antigens bound to their epitope can use their Fc receptor to bind to a phagocytic cell, macrophage, neutrophil. Some other cells can also be phagocytic. And so remember that opsonization is mainly just that the macrophage has an overall negative charge. The bacterium has an overall negative charge, so it's kind of tough for them to get close to each other. They kind of repel each other. That antibody just helps the phagocytic cell to get a little bit closer so that it can more easily phagocytose the pathogen. Okay, so we've mainly been talking about T-dependent antigens. So T-dependent antigens are antigens that need those helper 2 T cells to kind of produce the cytokines and kind of help to stimulate the B cells. So those are, for the most part, proteins. And so that's kind of the conventional antigen presenting cell, processes and presents the antigen. The helper T cells produce the cytokines, stimulate B cell activation. They go through all of their stuff and produce antibodies. But we also have some T-independent antigens. So, I said at the very beginning that the immune system really prefers proteins of a certain size, and that's the T-dependent antigens. But there are some pathogens that coat themselves with sugar, polysaccharides, with lipids, or maybe they produce things like endotoxin or lipopolysaccharide, and so those aren't as well recognized by the immune system as proteins, and so B cells can kind of do it on their own. They can B cells can directly bind polysaccharides, lipids, endotoxin, and kind of activate themselves. And those are called T-independent antigens. And so they, they help to kind of stimulate the B cell even without T cell help. So that's kind of a nice way that, the body can kind of cover more than just, proteins that are, presented by, like on a silver platter. B cells they're strong, independent women, and they can, they don't need no T cell help. They can do it on their own. So, okay, so those are called T-independent antigens. Polysaccharides, glycolipids, endotoxin, B cells can recognize them independently without helper 2 cytokines, and they can kind of go through the process even without the helper T cell help. Okay, and that just kind of shows the whole process. If you like pictures rather than words, I kind of do sometimes. Okay, and here's some more. The thing about these, independent, strong B cells that can stimulate, they can kind of motivate themselves. They can, respond to polysaccharide antigens and kind of, stimulate themselves to produce antibodies. It's not quite as good. So, they would be better off with helper 2 cytokines. They can do it, but they typically don't get too much past the IgM pentamers, so it's pretty good, but it's not great. Okay. All right. So I bet Dr. Johnson went through a lot of this, but the antibodies that are secreted are kind of serve as the feedback loop, and so we go through all this trouble of the somatic hypermutation, affinity maturation, and isotype switching, and all of that. And as soon as we get the mature antibodies, they go back and they start to inhibit the humoral immune response. And why do they do that? Because usually by the time we get that whole clonal expansion and there's a whole bunch of antibodies being produced, by that time the humoral immune system has kind of got the infection under control, and so those antibodies start to inhibit the immune system and kind of get it tamped down so it doesn't just kind of go wild and out of control. So antibodies kind of serve as a feedback loop to cause their ultimate demise. Okay, let's see. I think we're just about done. This is great. Oh, the other thing that kind of, so I can't remember if it was on that slide or not. The other thing that kind of causes a negative feedback is over time the antigens are just kind of depleted because the adaptive immune response does such a good job that it just, it just binds, neutralizes ADCC, phagocytose, whatever, complement, destroying all those antigens that it recognizes. So after a while, there's a whole bunch of antibodies floating around and they can't find an antigen to bind to because the antigens are all used up, they're already gone. And so that's another form of feedback loop. When the antibodies just can't find anything to do, then they kind of act as a negative feedback loop to kind of stop the adaptive immune response from going forward. Then those plasma cells that aren't needed anymore just die by apoptosis, save a few memory cells for the next time. Okay. So, and IgE, I just put this in here as kind of a special case. IgE, the Fc portion binds to Fc receptors on mast cells, and they are so they cause degranulation of mast cells, which we'll talk about here in a little bit as a manifestation of allergies. So whenever you if anybody has hay fever or allergy to cats or things like that, or molds or pollens, stuff like that, the antigen, which could be an allergen, will bind to the FAB portion. The FC portion binds to the mast cell, and that causes degranulation of the mast cell. And remember when we were talking about acute inflammation, we talked about mast cells when they degranulate, what do? They cause edema, they cause vasodilation, and so, and they also end up causing itchiness too. And so you think about if anybody has hay fever or allergies, when you are exposed to your allergen, you have, watery eyes, runny nose, itchiness, and kind of swelling in your respiratory passages. That's the effect of those degranulating mast cells that are causing that allergic or type 1 hypersensitivity response. Okay, just really quickly, this is the struggle between good and evil. So you can kind of see that the humoral immune system goes to a lot of trouble to try to fight off the bad guy that it recognizes. And so the bad guys are always trying to figure out ways to kind of one-up the humoral immune response. One of the ways they do it is that invisibility cloak, the polysaccharide capsule. But we see already that B cells have developed the ability to respond as a T-independent antigen to these polysaccharide capsules. And so, the B cells are winning out there. Another defensive way that bad guys try to one-up the immune system is they can go through some antigenic variation of themselves. So they kind of go through some mutations that to change their epitopes on their surface to kind of trick the immune system. And this actually is pretty effective. So have you ever wondered why you have to get a flu shot every year? Like, why didn't last year's flu shot work this year? It's because the flu virus is really good at changing their epitopes on their surface. Excuse me. So whatever humoral immune response you had last year in response to the vaccine isn't going to work this year because the flu virus has changed. Excuse me. It's just coronavirus, don't worry. I'm just kidding. It's allergies. So my mast cells are degranulating like crazy up here. So if you have minor changes, minor mutations in the virus from one season to the next, it's called antigenic drift. So it might just be a point mutation or something really little. Some pathogens go through a major change, antigenic shift, and so the more severe the antigen variation, the harder it is for the immune system to kind of recoup and adapt. So another defensive way that the pathogens can kind of evade the immune response is sometimes they can get inside host cells and kind of hide out in there, and the immune system can't see it. So like Mycobacterium tuberculosis, Listeria, Toxoplasma, toxoplasmosis, they can get in host cells and they can block the fusion of lysozymes with the phagocytic vacuole. And so they're kind of protected in air, and they're kind of invisible. Then there's some offensive ways that pathogens can affect the immune system. They can produce toxins, they can suppress the immune system, and they can make enzymes that can degrade the immune molecules. So they can do defensive as well as offensive ways to try to fight the immune system. But with everything else working well, if the patient is otherwise healthy, good nutrition, good rest, low stress, then the immune system is going to win out every time. Okay, so turning off the humoral immune response, we already talked about that. The mature antibodies go back as a negative feedback loop, and also when we run out of antigen, it kind of pushes the immune cells into anergy. Have you talked about anergy? Yeah, okay. So I always think about anergy as the opposite of energy. So energy is kind of, they hit the couch and they got the remote in their hand and they're not going anywhere. So they just kind of shift into neutral and they don't do anything. So they kind of stop producing the antibodies and they just become anergic. And then oftentimes, then they just undergo apoptosis after that. Question?